Imagine a community of people, all holding hands. They interact with each other, they talk, and they communicate. They are linked to each other, and this makes them a stable community. If one person were to isolate themselves from their peers, or conversely, become too greedy, they could potentially go on their own wild path to insanity. In our tissues, cells are also in a community. They hold hands and have their own roles and places in that community. This gives them their identity and their shape. This is what we call polarity. Similar to individuals in a community, if a cell were to lose its interactions with its neighbors, we would call this a loss of polarity. This step would make a cell more prone to become cancerous, as the proteins involved in polarity regulate essential cell functions such as cell proliferation, apoptosis, which is controlled cell death, and other critical processes. Additionally, if a cell is no longer able to control its division, and does so too violently through the uncontrolled upregulation of cell cycle proteins, for example, it will become cancerous. Our project aims to improve our ability in the early detection of cancer, in an effort to nip the series of events we have just described in the bud. And since early detection relies on the identification of particular biomarkers, we'll now turn to what molecules exactly are going to function as those biomarkers. Our research focuses on the interaction between circular RNAs, which I will refer to as circs, and microRNAs, which I will refer to as mirs. These are small, single-stranded RNA molecules. Sometimes they are released from the cell in packages called exosomes that then circulate in the blood, where they are easily accessible to us. Each particular circ is produced from a gene that codes for a protein. Mirs, on the other hand, are produced by transcribing certain parts of the genome that code specifically for mirs. Circs have the interesting ability to sponge mirs and limit their function, which is fascinating in its own right, but there's even more to it. Mirs have the essential function of gene regulation, being able to bind to certain parts of the genome and lower the expression of some genes. Now we see an axis is formed. We first consider a certain protein, encoded by a certain gene. This gene is up or down regulated as a result of certain external factors, which results in an increase or decrease in the production of circs. These circs, in turn, sponge mirs, whose job is to silence the respective genes. So the more circs there are, the less free circulating mirs, and vice versa. The less free circulating mirs means the higher the expression of the genes that they silence. This defined axis allows us to detect any modification in the expression of the primary protein, which is represented in yellow here, without the need to resort to biopsies. The extremely gradual nature of cancer initiation is the very aspect that makes it so difficult to diagnose and treat. A patient could have cells that are well on their way to becoming the villains we've described and could be completely unaware. There is therefore a large chunk of the population at risk of developing cancer, finding out only once they develop symptoms, at which point intervention through radio or chemotherapy is a must. Keeping all this in mind, it's easy to see why we think that early detection of cancer is a necessity in our fight against the disease. By identifying several axes of interest, we aim to screen for dysregulation in any one of them and quickly identify a potential predisposition for the development of cancer. The screening itself is done through a simple blood test, as the exosomes are already circulating in the blood. There even exist kits that help purify the exosomes and amplify the signals we're searching for. For example, focus on exosomes originating from breast or lung tissue. So instead of waiting until cancer can only be treated by chemotherapy or radiotherapy, both of which are extremely taxing for the patient, we can, upon detecting the dysregulation we talked about, promote an early change in lifestyle. This would obviously be a lot less tiresome for the patient. Going through chemo versus quitting smoking or changing diets, for example. It's not a very difficult comparison. Before taking a closer look at the axes we've put together, it would be useful to explain what the Kaplan-Meier, or KM, plot is. This tool allows for a quick and easy comparison between cases of high and low expression of a certain gene, protein, or mir. The graph is simply a prognosis graph, with one plot for high expression and the other for low expression. The source of the data is a wide range of published papers dealing with the gene, protein, or mir at hand. We note that we used KM plots for grade or stage 1 cancer, as we are focusing on the role of the axes in the initiation of cancer in the efforts of early intervention. One example of a well-defined breast cancer axis starts with SCRIB, a protein involved in polarity. Loss of polarity, as we said, contributes to the development of cancer, so the axis must logically start with a downregulation of script. Using a tool called circ interactome, we found the candidate circ associated with script, circ 0085872, that's logically also downregulated. Continuing the axis, and keeping in mind that the downregulation of the circ causes the upregulation of a mir, we were able to find, using circ interactome again, a mir that circ 85872 sponges, mir182. Here, KM plots came in handy, as we were able to clearly see how the higher expression of mir182 is involved in breast cancer. Finally, using a tool called target scan, we were able to identify an mRNA, and essentially a protein, that is sponged by mir182. This protein, GJA3, following the logic of the axis, is downregulated, something we confirmed using KM plots again. This makes perfect sense, as GJA3 is a gap junction protein, and is also involved in polarity. We now move to an axis relating to lung cancer, beginning with an upregulated protein. We will focus on the MYC gene, an oncogene found to be dysregulated in over 50% of human cancers. Due to the nature of the gene, it's an oncogene, it's only logical that the protein products, the MYC oncoproteins, are upregulated. Using the same in silico tools, we found one of the protein-associated circs, circ 0085534, that's also upregulated. This circ is known to sponge mir186, which is consequently less present in its free form due to it being bound to the circs. Finally, using target scan, we identified the target mRNA of mir186. The target mRNA encodes the CDK2 protein, which is involved in the cell cycle and DNA synthesis. Following the axis, the upregulation of circ 85534 would make free mir186 scarce, which would lead to an increase in CDK2 protein. Since this protein is involved in a very tightly regulated process in the cell, such an upregulation would cause the cell to become cancerous. The KM plot suggests that the higher expression of CDK2 indicates a much worse prognosis among stage 1 non-small cell lung cancer patients. Several factors can predispose a patient to develop cancer. Being an extremely multifaceted disease, there's a huge variety of lifestyle choices that could potentially put an unknowing patient on a collision course with cancer. For example, it has been established that obesity and excess body weight are associated with an increased risk for a wide variety of malignancies. This is partly due to chronic low-level inflammation and an altered gut microbiome, where a microbiome refers to bacteria in your intestine that are important for many bodily functions. For a country like the UAE, which according to some sources has a 31.7% obesity prevalence rate, such information is alarming. Thus, it is essential to develop programs for the periodic screening of at-risk populations to detect and intercept any cancerous growths early on, when the chances of recovery are much higher. Since the biomarkers that we rely on are in circulation in the blood, early detection could consist of, as we said, a straightforward blood test that would aim to screen for any dysregulation in the axes that we've talked about. We could even develop detection kits, further facilitating the detection process and allowing for it to be widely accessible and available. Cancer has long been humanity's public enemy number one, so much so that the phrase curing cancer has become synonymous with something impossible. It remains second only to heart disease in the number of yearly deaths it causes, despite enormous efforts to blunt the disastrous loss of life the disease causes. It is undeniable, however, that real and tangible progress has been made, and humanity is now in an infinitely better place to combat the disease than it was decades ago. We hope that the research efforts will add to that slow and steady progress, inching us closer to a time where humanity can finally say that it has indeed cured cancer.